which starts from the solid liquid phase separation, and then cell description method, and then concentration method, then final purification by using the chromatography techniques, then your exactly your formulation. These are 5 stages. So this is what we discussed in the previous class. Now I will move on to the biochemistry of fermentation. So we know that the conversion of the product is happening inside any of the fermentation process, but we should know exactly what is happening, what is have been converted into. So we know that there are different types of microorganisms, especially for the fermentation process. The main either the bacteria or yeast, or else it will also comes under the fungi. So fungi, all these can involve in it, and the fermentation process will be enhanced. So this fermentation can be classified into mainly two things. One is that we know that aerobic and anaerobic process. So if the organism requires oxygen, it is called as aerobic. If the organism doesn't require oxygen for its growth and division, then it is called anaerobic fermentation. So, so we can say like what is exactly happening inside it. Based upon also the microorganism, the conversion of your product will also be. So on using the glucose, we know that glucose will undergo certain reactions. The glycolysis process will happen, and this glucose will be converted into either any of the three components, either the we can say the lactic acid or else the ethanol component. If we are applying only the bacteria, the conversion of your glucose will be directly into the lactic acid. And if you're adding the yeast to your solution, sugar solution, then that will convert that glucose into a lactic acid and then to ethanol. Okay, this is what the overall structure of biochemistry of it. I'll tell you the equation exactly what is happening. So in glycolysis, from the name itself, you should know glycolysis means glyco means the glucose. Lysis means breaking down. So breaking down of glucose will occur in this reaction. Um, for example, this is C6H12 and O6. I think you all studied all these basic things in your 12th standard itself. So this will be on converting it. It will be utilizing one of the molecules, NAD molecules, and it will convert it to the pyruvate component. So pyruvate structure will be COO, that is C3 will be there, and H3 will be there, and O3 will be there. This is the pyruvate component structure. This pyruvate will further undergo on releasing the CO2 component, and it will convert it into acetaldehyde. So acetaldehyde will have a structure of CH3 CHO. Okay. This acetaldehyde on further conversion it will be into an ethanol. Okay. So this is what the production of ethanol. Lactic acid. So in lactic acid, what will happen? The glucose will be converted into pyruvate on utilizing the ATP, on utilizing the NADP, and it will release some of the energy, which is called as ATP, and it will convert that pyruvate into acetate, lactate. This lactate is also known as like acetic acid. So for these reactions, the whole organism requires some of the enzymes. We can say like in the ethanol production, the reaction requires pyruvate for the conversion of glucose to pyruvate. It requires pyruvate decarboxylase enzyme. And after the end of it, the aldehyde will be converted into alcohol. During that time, for the breaking down of your particular component, alcohol dehydrogenase enzyme have been used so that the aldehyde will be converted into alcohol. Okay. So while doing this process, the ADP will be also converted into ATP, which is an energy molecule, which will be utilized by the microorganism itself. Okay. Yeah, you can see this reaction. Glucose, lactic acid, then you will get also ethanol and carbon dioxide. We have to filtrate. On downstream processing, we will filter the lactic acid alone and ethanol alone by using any distillation process, no, by varying, because lactic acid have a varying boiling point, and the ethanol will have a varying boiling point. So based upon that, we can separate it both alone. Okay, this is for the lactic acid. So for the conversion of pyruvate into lactate, it required lactate dehydrogenase enzyme, which is more important on converting and giving one of the hydrogen to the NAD, and this NAD will become NAD plus. Okay, this is what the steps exactly happening. Glucose, you know about the cycle, Herb cycle, and also glycolysis process. Glucose will be converted into glucose 6-phosphate, glyceraldehyde 3-phosphate, and then it will reaches on further steps. It will reaches the pyruvate. Then this pyruvate will be converted on the presence of this lactate dehydrogenase enzyme. So, what are the basic fermentation methods? We can say like. For the raw material like milk, it will be fermented and it have been converted into yogurt or cheese. And for fruits, we can say we are getting the vinegar. And for barley, on the conversion of fermenting, we will get beer. On the vegetables have been converted and it have been fermented and it will become a product kimchi, and milk will be converted and it will be having an enhanced nutrition. We will get a different flavored and enhanced yogurts will be present. That will be examples of. So kimchi is an yeah, here the for the vegetables they have went for this fermented product kimchi. So it is nothing but cabbage have been submerged in brine. Brine solution, you know that the salt solution. So in that, what will happen is that the lactose present in the vegetables will, and also the sugars present in the vegetable will be get fermented and it will converted into a lactic acid. So this food will be very tanky, and it is a fermented food. It's maybe you're using in the east side of our country. Then yogurt, which is what we are having in our house. So curd, we can say as yogurt.